Welcome to experiment on sugar analysis using the 3,5-dinitrosalicylic acid methods or DNS method. In this practical, we will learn how to measure reducing sugar like glucose using a color change reaction and a spectrophotometer. Carbohydrates, especially reducing sugars like glucose and maltose, play a vital role in biochemistry. What makes them reducing is the presence of free aldehyde or ketone groups, which allow these sugars to act as reducing agents in chemical reactions. Measuring reducing sugars is important in several fields, including metabolism studies, where they help us understand energy use in the body, food chemistry to monitor sugar content and food quality, and fermentation analysis, especially in processes like brewing or biofuel production. In this practical, we have four main objectives. First, to introduce students to the use of spectrophotometry in preparing a glucose standard curve. Second, to demonstrate how spectrophotometry can be used in the quantitative analysis of reducing sugars using the DNS method or 3,5-dinitrosalicylic acid method. Third, to guide students in preparing a series of glucose standard solutions, which are used to construct a calibration curve. And finally, to determine the concentration of a known sugar sample by interpolation using the absorbance values plotted on the standard curve. For the experiment, the following materials and equipment are used: a spectrophotometer to measure absorbance, a hot plate for heating reaction, several glass beakers for preparing solutions, cuvette for placing sample into the spectrophotometer, test tube and a test tube rack for organizing and holding samples, micropipettes and tips for accurate measurement of liquid, distilled water as a solvent and for dilution, a standard glucose solution with a concentration of 10 grams per liter, sodium-potassium tetrate, also known as a Rochelle salt, and dinitrosalicylic acid or DNS reagents, which react with reducing sugars to produce a measurable color change. For method, pipette duplicate the following reagents into a series of tri-clean and a label test tube as indicated in the following table. 2. After replacing the above mentioned solution in the label tube, shake well and then place them in a boiling water bath for 5 or 7 minutes. 3. Cool the tubes at 1 ml of Rochelle salt. Then, transfer the solution into cuvette. Read the extinction optical density of the color solution at 575 nm using the test tube 1 as a blank. Before we start to the next step, we must make sure all the tube must be cooled to room temperature before reading, since the extinction is sensitive to temperature change. Record the reading and plot the relationship between the optical density and the concentration of glucose solutions. See whether there is a linear relationship between the concentration of glucose solution and their corresponding optical densities. The last one is keep the standard curve to determine the glucose concentration in the selected sample that will be used for the practical 4. its result for this experiment. In summary, the DNS method is a simple and effective way to measure glucose level. A standard curve was made using known glucose amount, and darkest colors show a higher sugar concentration. The absorbance was measured at 575 nanometers using a spectrophotometers, and the results were consistent. This experiment helped students practice key lab skills like using pipettes, mixing a solution, and reading absorbance from a standard curve. These methods can be applied in many areas, such as food testing, health checks, and fermentation processes. Through these experiments, students also learn how chemistry connects to biology and how to measure sugar in a known sample using a standard curve. Hi, welcome back. This is Practical 2B, Spectrophotometry, Determination of Sugar in Sample using the 3,5-dinitrosalicylic acid DNS method. Introduction. Measuring sugar concentration is important in biochemistry, especially in food testing, fermentation, body fluids. Reducing sugar like glucose, maltose, and lactose have a aldehyde or ketone groups that make them act as reducing agents. Accurate measurement of reducing sugar help in understanding metabolic pathway and ensuring quality control in food and medicine. The DNS 3,5-dinitrosalicylic acid method is commonly used to detect reducing sugars. In this method, DNS react was reducing sugars at high temperature and it's form a reddish brown color. The last one is the darker the color, then the more sugar is present. For objective, first introduce to the student to the use of DNS method for quantifying sugar concentration in a given sample. Second, to introduce the DNS method as a colorimetric technique for quantifying the concentration of the reducing sugars is a known sample. 3. To emphasize the importance of accuracy and precision in laboratory-based sugar estimation procedures. 4. To develop student ability to apply spectrophotometric measurement in biochemical analysis. For material, first, we use the DNS reagent, Rochelle salt, cuvette test, tube and test tube rack, hot plate, glass bakers, distilled water, micropipette and tips, selected drink or sample. For methods, first, prepare clean and dry test tube. Second, add 1 ml DNS reagent into all test tube containing samples as a measure according to the above table. Third, boil the test tube for 5 or 7 minutes. 4. After boiling, let the test tube cool down to room temperature. Add 1 ml of Rochelle salt solution into the mixture. The last one is the color development of the mixture is then read a 575 nm by using a spectrophotometers. Test tube is the blank set as negative control for the eliminate background readings. our result for determination of sugar in sample using the 3,5-dinitrosalicylic acid or DNS method. For conclusion, this experiment shows how the DNS method works to measure reducing sugar like glucose. A glucose standard curve was built using a known concentration. Absorbent was read at 575 nm as a known sugar level were calculated using the curve. The curve shows a strong, reliable correlation between sugar concentration and absorbent. Result match the expected dilution pattern, proving that color intensity increases with a sugar content. This matches the theory reducing sugar have a aldehyde and ketones group that reduce DNS and form color. Student practice important lab skill, pipetting, making and diluting solution using a spectrophotometers and analysis data. The experiment show that precision and proper technique are very important. Issues like uneven heating, incorrect timing, dirty glassware, poor calibration, and can affect accuracy. The DNS method is also useful in real life such as food testing, fermentation monitoring, medical sugar testing. This experiment helps students build both scientific knowledge and lab skills that are useful in research and industry. Thank you for listening. This is our experiment practical 2A and 2B. Bye-bye.